Now audio. Tree branches down the zip. All right, good morning, everyone. Thank you for joining, especially all those on Zoom in different time zones.
The volume might need to be up a bit if you can control it.
Can I? I have no idea how to do that here. What if I speak louder and stand closer?
That's
Okay,
fine.
better. Good.
You're just too tall.
I don't have that.
Sorry about that. I'm very happy to welcome Beth Simony today. Beth works at the Broad Institute, but before she started working there, she actually started here in Boston at the Boston University, did her her bachelor, different university system than in the Europe. Then moved to California, San Francisco, and did her PhD there. And then returned to Massachusetts, to Boston, to work with Ann Carpenter. And then sort of, I wouldn't even say worked her way up through the ranks, because it's been pretty fast, and she now runs her own lab of currently a perfectly gender balanced 4 of each, meaning 8 in total plus Beth, image analysts. And as far as I can tell, Beth is everywhere. She's at every meeting. She's in every opinion piece. She's organizing things. And this is also a harvest year. If I just check Google Scholar, I think we're counting 27 publications this year. So yeah, you make us all look bad. Thank you. Apart from that, she's also highly organized. Maybe that's the secret to actually being so successful. And I would also highly recommend, if you run a lab or want to run a lab, take a look at her website. There's some very good instructions on just ethical behavior and tech setup and what people can expect from you and what you expect from them. But enough about that. Beth, please, thank you for coming.
All right. Thank you so much for having me all the way from long and far across the river. But no, very, very excited to be here. I was so excited when I heard that Simon was going to be potentially coming to HMS, because I've adored working with him everywhere our paths have happened to cross, usually at New Bias before this. And so very much looking forward to helping make Boston sort of the nexus of bioimage analysis within the US, certainly, and pulling together a lot of really interesting things, because it's a very exciting time to be in bioimage analysis and to be thinking about how we use microscopy data actually as data. So I'm part of the Broad Institute imaging platform, again, located all the way across the river. Ann Carpenter, who's our senior director, started the platform in 2007, so we've been around almost 20 years now. She and her co-PI Shantanu Singh now focus on sort of the image informatics of bioimage data. I think you guys can see my mouse, yes. And basically, once we turn images into data, how can we do new kinds of question answering, and how can we design new kinds of assays? My lab works a little bit upstream of that, which is just how can we make new image analysis software and make image analysis workflows so we can help people turn those images into data in the first place, and not just any kind of data, but the right kind of data. So I'm going to tell you 3 short stories about, first, how can you just analyze lots of images without learning to code? Nothing wrong with learning to code, but sometimes it's not what you want to do right now. And then what if you want to do that, but also with deep learning? You've probably heard about deep learning somewhat over the last couple of years. And then finally, sort of how can we then even take this a step further, and in a way that does require some code, but use more of the pixels in the images and sort of really transform the images into the truly high dimensional feature space that they always sort of were, but just hiding pretty well. And I like to start with this picture just because it really sort of emphasizes just how much information we can get from biological images. So this is just from the cell painting assay, which is an assay where we're looking at different organelle dyes. And I'm going to talk a lot more about cell painting in the sort of last of the 3 stories. But imaging has a lot of value that even as techniques like single-cell RNA-seq have come on the field and exploded and are doing things that we never thought were possible 10 years ago, the fact that with a microscope, we can get information about molecular interaction at a single cell level very easily, and see how different things are happening within a cell in terms of behavior is still something we can't do, certainly for the cost at the scale with any other sort of omic technique. But because there is this historical artifact that microscopes came around long before computers did, we still, a lot of times as a field, think of microscopy as qualitative. I am very glad I was not doing microscopy in Ramon y Cajal's time, because I cannot draw this well. And if you do ever get a chance to go see his art, it is truly lovely and it is truly art. But we don't necessarily at this point want to be doing art, we want to be doing science, and those things overlap. They're not entirely distinct, but they're not entirely the same either. And I'd like to say we've gone totally beyond that, but we haven't totally. I'm going to tell you just one sort of very short story from my time at BU, Go Terriers. Not because I think it was actually that unique of a story, because I think, you know, again, this is 15 or so years ago. It's pretty common of a story, and it's still not that uncommon of a story today. Representative image shown. So here we were looking at salamander retinas and trying to look if the signaling molecule that is in the green cells is talking to and making a downstream signaling molecule that is in the purple cells. And certainly it looks qualitatively like the green cells are talking to the purple cells here in these process layers in our retina. So representative image shown, they're clearly talking to each other. We solved it, right? And so when I asked my boss how to prove this, he told me to print them all out, put them on the floor in piles by treatment, and then figure out what conclusions I could draw in terms of whether particular treatments cause changes in this particular molecular interaction. And again, I think this was pretty typical, and I think this is still what happens in a lot of labs. We did a tiny bit of quantification. We did some line plots, and hey, look, it looks like the green and the purple are talking to each other. But it's still really thought that microscopy should operate this way and does in practice operate this way, which is really a shame, because the digital camera exists now. And when we are looking at these digital images, they are arrays of numbers, and we're not used to thinking about them that way. Whether it's in this toy example or whether it is in this picture of a man taking a picture. When you take pictures on your smartphone of you and your friends out at the bar or whatever, these are ultimately arrays of numbers. And so if you think about it, if your phone has a 10 megapixel camera or your microscope has a 10 megapixel camera, that's 10 million data points every time you snap that camera. And you might snap it for multiple time points or Z-planes or channels. And so you're gonna quickly get into the billions and trillions of pixel data. Which is a tremendously rich data source. And, you know, as data scientists, we should be really excited about that. And this paper is about 10 years old, but I'm not sure how much it would change if you ran the same thing today. It might be kind of interesting to redo this study now, but looking at papers who report themselves doing high content microscopy, which again, should be the microscopy with the most data science sort of attached to it. The vast majority of papers only reported 1 or 2 cellular features that they measured in those millions and billions and trillions of pixels. So there's a lot of data that's still really just being left on the table. And so the sort of reason that the imaging platform exists is to actually sort of turn those images into answers. So, okay, you're like, yeah, whatever, this, you know, I see my phenotype just fine. Your purple and your green cells were clearly talking to each other. You know, why are you going on about this so much? So there's one problem about thinking that you can see your phenotype just fine, and that's really just that the human brain is a bad quantification engine. When I first put up this picture, or possibly if you're looking at this on Zoom on a pretty small screen, even still, this looks like a color picture. This is not a color picture. This is a grayscale picture with some lines drawn on top of it. And your brain very helpfully fills in the missing information. Because your brain is designed to help you find things around you quickly, which is great for a lot of things. It's bad for quantification. And so your brain is constantly lying to you about the world around you. And so it's not really the tool that we want to use for quantification of our phenotypes. So I'm going to talk about 2 different ways of thinking about images. In the first 2/3 of the talk, I'm going to talk about ways that we measure and score known phenotypes. I just said to you, your brain is lying, but it is still probably one of the best neural networks on the planet. So we do want to use our brains sometimes if we think we have other good ways of telling what's going on in an image. And then in the second part, I'll talk about how we do some things around unknown unknowns. But still, the vast majority of biology is done by looking at images or looking at the field and coming up with hypotheses and then seeing if we can design microscopy experiments to validate them. I can vouch for the fact that this was going on well into the aughts and the tens because I spent a lot of my graduate career fusing GFP onto my favorite proteins. And this is, again, how a lot of amazing biology and amazing discoveries get made. So we don't want to say that this is bad to do. We just want to say that we want to make sure that we're backstopping it carefully with image analysis. And so I'm going to start by some high-level prescriptions of when you are making an image analysis workflow, what you should do, because then I'm going to talk about different kinds of technologies you can use to build these workflows, but the principles are really the same no matter what you're doing technologically. So most image analysis workflows, though certainly not all, start with finding an object that you care about. I'm probably just out of force of habit going to say cells a lot because usually when I'm talking to the people I particularly work with, we're looking at cells, but you can feel free to substitute organelle, C. elegans worm, whatever is the object that you care about within an image. And when you are finding those objects that you care about, you have done what we call segmentation, or if you're a computer scientist, instance segmentation specifically, what you should actually do when you've gone through and you've created a workflow that does that instance segmentation, which is typically the really hard part, you should ask yourself, is it generally right? If it's not generally right, there's really no point to going on to any of the rest of the questions. In cases where it's detecting too much or too little, do we have about the same amount where there is too much detected versus too little detected? If it's in one direction or the other consistently, you can probably tweak that away, but otherwise there's probably not much you can do. Likewise, when it's then actually breaking the stuff in your image into individual discrete objects, is it about equal numbers of times sort of saying that two objects that are touching are one object when they're really two versus splitting an object in half? And then most importantly, almost never in biology are we trying to look at a single sample. We are usually trying to look at some sort of a treatment or a gene change or some sort of paired samples or many samples. And so do all of those things hold true across the most extreme expected phenotypes I expect to see in my samples? Take it from me, there's nothing more heartbreaking than spending months chasing a really exciting change in measurement that turns out to be a segmentation error. It's not fun that day. And then you have to go to your boss and be very apologetic about all the money you spent on these experiments that turns out were just a segmentation error. The tool that we generally recommend people get started with tapping into those arrays, especially if you're not somebody who's, again, super into code, is a tool called Cell Profiler. Ann Carpenter and Ray Jones wrote the first version 20 years ago this year, so happy birthday, Cell Profiler. I stumbled across Cell Profiler around here in graduate school. You can see people have been working on it a lot since then. I joined the team, sort of more in the here-ish range. But I mostly want to point out, the amazing team of people who've worked on the software over the course of 20 years, and this is a tiny, tiny subset of the larger number, as well as point out that I think part of the reason that 20 years later Cell Profiler is still around and still relevant is we've always had on the development team a mixture of these people who are highlighted in blue, who are the people who are computer scientists and software engineers by profession, and the people who are highlighted in green, who are people like me, who are biologists originally by training. And so by mixing those groups of competencies in making the software, we have good software development practices, but also all of the documentation and all of the structure of the program is designed around the way biologists tend to think about image data. Just some stats for those of you in the room who are software engineers and might think some of this is cool. So Cell Profiler has about 94 different modules, and I'll talk about what a module is in a second from a sort of functional point of view, but essentially an image analysis operation of some kind, which might under the hood be many mathematical operations. And we spend a lot of, Simon pointed out our website has a lot of documentation and things like that. A lot of that comes from Ann Carpenter. I can't really sort of say that this was my personal way of doing things at first, but she has definitely converted me over to how important the documentation is for actually making sure that people can use the thing that you're trying to make. And so Cell Profiler's code base, depending on exactly how you count, is about a quarter documentation. And it needs to be well documented because we have at least 2 different user bases and then people all along the spectrum. But roughly speaking, my very unscientific estimates, About 90% of the people who use Cell Profiler have never opened the terminal. They never will. Once you ask them to, they are going to leave the room. And then about 10% of our users are people who are doing this at universities or at biotechs or at pharmas and who want to run this on millions of images on their HPC at scale. And so we have a lot of fun trying to satisfy both of those clientele. But so we need to have something that sort of adapts to both user friendliness, but also to being able to make reproducible workflows. So this is the Cell Profiler interface. This isn't a Cell Profiler talk, so I'm going to talk about this only for a minute or two, but just so that I can give you a sense of what is this tool that we make. This is ultimately at its heart an image analysis workflow tool, and the magic is here in the what we call the pipeline panel. So Cell Profiler doesn't really contain anything that any other open source image analysis software like Fiji or QPath or anything like that doesn't also contain. But what it has is a workflow engine, essentially, that you can use to take those 94 modules, which might be something, I said I'd come back to those, like cropping or finding objects or measuring objects or saving things. And you can mix and match them in a reproducible way in any order that you want with any sort of amount of configuration that you want. Individual modules might have only a couple of settings that you can tweak. The images module here, which is where you drag and drop your images in, has very few configurable settings. Some of them have, in the dozens. And ultimately what this allows you to do is, again, in a way where you don't have to learn to code, you can make image analysis workflows very reproducibly. And then you can, this creates a plain text file. So you can attach that as supplemental information on a paper. You can give it to your lab mate and say, hey, this is how I did my analysis. You can do yours the same way. And it just makes it easy for your image analysis to scale to however many images you have, whether that's 5 or whether that's 5 million. The most important button in Cell Profiler is the help button. I mentioned that we have 94 modules. The settings count is close to 1,000. That's a lot of stuff. There's still some things that I wander across in the code base. I'm like, wait, we do that too? And so every single one of those 1,000 settings has a help text written by a biologist who actually can then help you figure out what is that setting doing, what is that module doing, how should I set it based on what my images look like. And again, computer science maxim of powerful, user flexible, easy to use, pick 2. We're trying for at least 2.5 by at least sort of documenting the crap out of this thing. There's a test mode where you can play around with settings, and then once you've tested your settings, you can set the output and go to analysis mode. If this were Cell Profiler talk, I could go on for quite some time, but I'm going to just move forward here. I mostly just want to show this slide because I want to tell you the hardest part about Cell Profiler is getting it started. And in that sense, I mean getting the images loaded in and doing the bookkeeping of what your experimental setup is. Again, part of the challenge of making something that is really designed to be flexible and sort of work for any use case is you need the user to tell you if this is, 150 channels because this came off an imaging mass cytometer, or this is one channel, but 5,000 Z-planes because we're doing something where we're looking at a whole huge volume, or whether this is many time points because we're doing tracking of something. And simply, you need some way to actually tell Cell Profiler what the experiment is. And people like to name things and structure things in a wide variety of fun layouts. And so this is usually where people fall off. I just want to point out, my team put together a really great blog post along with video tutorial with sort of the 10-ish most common ways of configuring these input modules to do all the bookkeeping. And so if you go to that link, and again, all of these, the slide link is on almost all of the slides, that will help you get past this initial sort of headache of getting the data in. And from there, it's relatively straightforward. So if this were my Cell Profiler talk and I was about to start teaching you classical segmentation, oh boy, there's a lot of slides here to teach you guys how to do classical segmentation if you've never done it before with using classical computer vision techniques. I did stick them at the end of the deck. Again, if this is something that you actually want to learn, we also have a ton of YouTube videos on this. But that's kind of a lot of slides. You guys don't want to learn all that. I don't really want to learn all that either. Can we not? Can't we just design something better that doesn't require quite so many slides? So deep learning. you've probably heard about this somewhat. Chat GPT and, DALL-E and all of the amazing things that deep learning can do. It's really become something where it's gone from something that like 20 computer scientists over in the corner care about to something that is touching every part of day-to-day life. And that includes microscopy. There are a lot of things that you can do now with deep learning around doing things like correcting an image, or even, asking an image, hey, what would this staining have looked like if I did it, but I didn't? Getting out image-based features or doing activity prediction. I'm going to talk about that sort of in the very last part. But really, where one of the first use cases where this has become really popular in bioimage analysis is in this cell segmentation. And so these are some papers from us, some papers from other people, but just sort of looking at the wide variety. And this is a tiny subset of all of the many things where you can stick in this array of numbers and get out answers. Yes.
Some of what you showed right here, including segmentation, can be done with canny edge detectors. This is not necessarily deep learning. So what's the actual usage of deep learning, the deep neural networks to this process for making it more information rich?
That's a great question. So the question specifically around segmentation is you can do this without. Part of it is simply that using neural net. So again, you can do segmentation with classical computer vision. It's just non-trivial. Whereas a network, you can sort of train with parameters in ways that tend to be easier, and the user doesn't have to know quite so much about what each individual parameter does. It's not necessarily better in terms of the, if we were looking at the F1 of the, ground truth to the output, it's just easier. But that's a great point, and thank you for emphasizing that. Awesome. Please do feel free to keep shouting out questions. I love when people do that. And so again, deep segmentation is really probably where we've had the deepest penetration in this space so far, because segmentation is hard and people don't like setting it up. And so if we can come up with nice, easy ways to do it, then people would rather do that. And so Stardust has been around, I think, about 5 years now, which was one of the original ones that really sort of offered a nice user interface for doing segmentation using deep learning in a Jupyter notebook, and is still a fantastic tool and sort of state of the art for a lot of problems. Next on the thing was CellPose, where we are computing these object flows via a UNet and then turning the object flows into instance masks downstream. For both of these, I do want to point out there is a Cell Profiler plugin available. So if you would like to access these sorts of segmentations in Cell Profiler, you can still get the benefits of a text-based workflow tool, but also the benefits of deep learning. So great. All right, that problem solved. Not quite. So here's the problem. Scientists who working on turning images into data, who if we ask them on a scale of 1 to 7 to describe how much they spend their time doing imaging versus image analysis, cross a wide variety of things. And then if we ask them how comfortable they are developing new computational skills, how much are you willing to learn new compute? There's a pretty wide variety. It might not surprise you to know that people who spend most of their days on their computers are more comfortable learning about computers. This is not, maybe the world's most surprising finding, but it means that, again, especially if we want to help people who are at the bench doing these imaging tasks and then want to immediately turn them around into answers to their biological questions, we need tools that cover a wide variety of computational comfort levels. And deep learning is just unpleasant to install. It really just is. Even, again, this is not that my team is bad at their jobs. They're fantastic at their jobs. It's just a hard problem, especially when you're talking about across tons of OSs and, somebody's got a computer from 2010 and all sorts of things. And this is really a problem. So when we did a recent survey of the image.sc forum, about 1 in 5 threads have install somewhere in the thread. About 1 in 10, it's in the opening post. So about 1 in 10 posts are sort of being opened up, possibly due to an installation issue. This is not a small problem, and it's only going to get worse the more of the field moves to deep learning, because deep learning is unpleasant to install. What can we do to sort of try and solve this? So one way of going, and I think it's a really powerful way of going, is containerization. So if you're not a software engineer by training, containerization, you can think of it as an OS in a box. It's not 100% accurate, but it's close-ish enough to accurate that you can sort of think about it that way. But the major thing to think about is that installation happens once and only once. So rather than if you want to use Cell Profiler and you want to use Cell Profiler and you want to use Cell Profiler, that's 3 installations. It's 1 installation, and then all anyone has to install is a container runner, and then all of the actual compute happens inside the container, where again, our tool and our data have only been put there once. I'm not going to read you this whole slide, but in general, this is great also because it's, you know, it's pretty common now in sort of other parts of scientific software, such as a lot of bioinformatics. Biocontainers has more than 1,000 different Docker containers that are out there. I say Docker just because, again, in the same way you say Kleenex, it is the sort of one that is the most common, but a lot of Docker containers can be run in Singularity, and now there are things like Podman that you can use to run these containers. But this is potentially a really nice way to get around the installation burden. And so image analysis is working on getting towards this. I forgot to update this. This is now published. Where we are now starting to actually say, hey, look, this is a checklist for publishing image analysis workflows, and we are, this was a group called QUAREP, the Quantitative Reproducibility of Light Microscopy is QUAREP LIMI. And this was their recommendations that were recently published in Nature Methods around when you are publishing microscopy experiments and image analysis experiments, what should you actually have to include in your publication? And so we are pushing them, especially for machine learning workflows, to include containerized versions of analyses, because it's simply the way that's going to be the most reproducible. It's not perfectly reproducible, nothing is, but it's the closest we can get right now, short of shipping somebody a laptop, which is one of the things that the Napari folks have joked about in terms of like the most reproducible compute thing is you just stick a laptop in the mail. And there is this bias that that containerization is inherently unuser-friendly, and I'm happy to tell you that's simply not true. So here's a 1-minute video in which I'm going to look for a software container that contains, in this case, just sort of Atsu thresholding and watershed. So not a neural network, but I can tell you we've successfully used this approach for neural network image analysis tools as well. And without touching my command line, so all of the people who are command line allergic can use this just as well. I tell the Docker desktop program to give me a particular image analysis container and to run it in a web browser. And again, in under a minute, I get from looking for the thing that I want to find to giving it some images. I have some tunable parameters here. I'm not going to tune them just, you know, in this one little 1-minute demo, and then download my results. Again, this probably wouldn't be quite this fast if this were truly running a deep learning workflow, although, you know, inference can be fast. We're not training here, we're just inferring. But there's absolutely nothing unuser-friendly about a container if you build it the right way. And this is the entirety of the code for that container. You can find it on GitHub. The Docker file itself is 20 lines of code, and the app file is something like 80. And even something like Cell Profiler doesn't actually need that fancy of a Docker file. This Docker file for Cell Profiler is 24 lines. And so this is something that we're really encouraging people developing image analysis software to do. Yeah.
I don't know how it is at Broad, but sometimes we have a lot of issues with containers because we have IT security doesn't like it. For example, this radio file won't work because radio is blocked on our network. Is it just simply an institution thing that you find, or do you think that there's other ways of doing that won't get you in trouble with IT?
It's a great question. So around, again, if you can't hear online, around security and IT potentially blocking some things. I've never heard of IT blocking radio before. That one's new to me. Certainly, Docker is not a great fan, or does not have a ton of fans in sysadmin because it does have root privileges. Singularity can run a lot of Docker containers, which helps with at least some things. But yes, it's definitely very institution dependent in terms of what your institution will allow you to do. But that is a fantastic segue into well, why don't I just skip the slide so I can go with the fantastic segue, which is, okay, so Docker containers, that's one step easier. Can we get easier? Now, that person had to install Docker Desktop. Maybe Docker Desktop is blocked at your institution. What else you got? What if we just stick everything in the web? What if instead there's no installation at all whatsoever? And we just put everything in a web tool. This, doesn't solve every problem, but it solves a lot of problems. And so for a few years now, what was originally a collaboration with the Carpenter and Horvath groups, and now is sort of continuing in my group, we've been trying to make this deep learning web tool for scientific images. The major tool, Web Tool Pro, is no installation, and you can run it anywhere. The tool I'm about to show you, I have run on my phone, my Kindle Fire, people run it on iPads. I've seen somebody do it on a Steam Deck, which I didn't realize it could do. But the web gives you things like accessibility, because really smart people at Mozilla and Google and lots of places have spent a lot of years working on that. More software engineers than I personally have the budget to hire. And the browser can take things now with things like WebGPU of like accessing the GPU. And so if you have one available to you, great, you can use it. But if you don't, that's fine too. Now, is a web tool going to be powerful to do what we want? Not just in what's a client server model where we're uploading our data to the internet, but locally. The web is, unless you're using something like WebAssembly, if you're trying to use actual JavaScript and TypeScript, you don't really have the library support. There is no skimmage for JavaScript, sadly. And you need internet access or to be able to run some sort of a container. And so the tool that has come out of all of this work is called Piximi. You can go to piximi.app on wherever fine web browsers are sold, and you can try it out yourself. So again, in our experience, people who care about images either care about whole images or about objects within them. And so in order to sort of get at what users care about, they probably care about classifications, measurements, or segmentations, and so that they can make classifications or measurements of objects. And if we want them to be able to make segmentations, we probably need them to be able to make annotations so we can do human in the loop segmentation, like Cellpose 2, for example, has shown to extremely great effect how a human in the loop segmenter for deep learning that a user can train can completely revolutionize a field. If you haven't tried Cellpose 2, try Cellpose 2, it's really good. And so this is what you'll see right now if you open up your web browser. You can look at images and you can look at objects, which we call annotations, because ultimately that's how they're made, at least for now. And there is a friendly web browser that you can look around in. If we look at the annotations, we have this nice individual image level viewer in addition to this experiment level viewer that we have here. And the annotation tool has all of the fun things you might like bounding boxes and a pen, but also some nice things like super pixels. This is my favorite. This is the color annotation tool where you just click and drag and then hit enter, and then hotkeys, you're sort of through annotating your image in just a very short amount of time. Piximi has pre-trained object detector and segmenter models in it. So if you were at Elmi this year, this is what you saw at the beach, and then this is what Piximi saw. It found some people. It didn't find everybody, but that's Coco's problem. That's not our problem. And here I'm going to actually show you, in this case, running Cellpose. So one thing about Piximi is that all of this, with the exception of exactly what I'm about to demo for you right now, is happening client side. This is happening locally in your browser. Once you have your data and your network loaded, you can put your computer in airplane mode and it will work just as well. We are not taking your data. We do not see your data. This is absolutely all happening client side, with the asterisk of if you want to run Cellpose, it's happening at the bioengine in Sweden. And so that we have to send out elsewhere because Cellpose is in Torch, not TensorFlow, and this is TensorFlow.js.
Nice.
Simon.
So we have a question from Slack. I don't know where the people on Zoom, sorry, Zoom. Somebody's asking how to deal with privacy concerns. So if you have patient data and you want to analyze the data on the web, how do you deal with that?
Yeah, that's a great question. One of the ways you do it is by not having the data ever hit the internet. You can open Piximi and then you can put your computer in airplane mode, and then you don't actually ever have your data touch the web. You can run Piximi out of a Docker container. I just showed you an example of a Docker container spawning a web browser. You can do that with Piximi, and then your data has literally never hit the internet. You can literally have the computer never have been connected to the internet in its life. But yes, absolutely, when you start getting into things that run client server, like the Cellpose thing I just showed, or like a lot of things like the bioimage model zoo and deep cell kiosk and a lot of those things, you have to be very careful, and you should follow your country's relevant laws and things, which depending on which country you're in is very different, and I can't advise you further beyond that. But it's a very good question. That's part of why we built this client side. So thank you for that. Great question. This is my favorite one. This is the classification tool. So we are actually going to fit a classification network. This is real time. Here we have cells that have either no GFP, cytoplasmic GFP, or nuclear GFP. And we are going to fit a classification network in my web browser right here. Again, this is real time to actually learn the difference between those three classes. And then we can do things like look at the confusion matrix, we can fix mistakes, and we can retrain if we need to. And there are hyperparameters that are hiding there. So if you are a person who knows what a hyperparameter is or wants to learn what a hyperparameter is and wants to learn how to tune things so that your neural network can be better quality and more efficient, you can do that. But we've made it friendly so that if you don't, and if the idea of hyperparameters makes you want to run from the room, you never have to actually look at what those are. And it does work on your phone, although I've been reliably told if you pull it out on your phone at a bar, it makes you not cool. I don't agree, but that's what I've been told. I mentioned before about reproducibility and automation. We haven't forgotten about that with Piximi. Again, almost everything is client side. If you are running server side, it does warn you. But again, you can serve it from a Docker container, so you can do this entirely without the internet. We're not taking your data. We've never seen your data. We're using things like ZAR formats, and there is a project file that you can say, here are my images, here are the annotations I made. Hey, friend, go look at this. Or hey, journal, take this, and this is my supplemental data. It's actually all of my data in my analysis. It makes it easy to reproduce. And we're using standard TensorFlow model formats. There's a very easy conversion to take a TensorFlow.js model and turn it into a standard TensorFlow model. So you could take 1,000 images, train this in your web browser, and then export the model, export the weights, and then move to the million images that you have in your cluster or cloud somewhere. Segmentation of models in app isn't happening yet, but we have very clever people working on how to do that efficiently enough that it actually works in your web browser without trashing your whole computer. And then the last thing is going to be measurements. And so we have some of the measurements built in now. It's looking really great. And so the idea being that you can get from your images to the answer to your question about what's going on in your biological or whatever kind of science you do, images, without ever having to leave your web browser. We are developing this live and in public. It does mean occasionally run into bugs. Please report them if you find them. But we are always happy to work with other people and keep stopping by for updates. And there will keep being more and more that you can do in this tool. So moving into the last part of the talk. So I've been talking about things for finding objects for the most part and things for sort of basically, yeah, doing segmentations. So what? Why? So software can help us do lots of metrics. And again, I started with this idea of millions and millions of pixels. Let's double down on that. Let's turn those millions of pixels into at least thousands of measurements. Let's not stop at one or two. And so when people think about image measurements, there are some things that come to mind and that make sort of intuitive sense to human brains. Things like intensity. If you're doing fluorescence, you might say, what is the mean intensity of some marker inside some object? Maybe the object is part of that marker, maybe they're different. But again, the GFP intensity inside my nucleus, I just showed an example of where that is a biological phenotype of interest. Or the distribution of that intensity. If you want to get a little bit fancier, you can do things like, turn the cell into concentric rings and then say how much is it evenly distributed within and across the rings. Size and shape is one of the other ones that people are pretty used to thinking about from the point of view of a biological measurement. And something that if I say to you, your cell has a diameter that is twice as big, you probably get a mental picture in your head of how that object has changed when the diameter gets twice as big. Colocalization, I forgot colocalization comes after size and shape in this slide deck. But so here's the thing. I started also with the idea that human brains are crappy quantification engines. And that's true. So here are cells from an imaging flow cytometer in different cell cycle phases. Who wants to come up and tell me what they all are? Nobody ever says yes to that. I finally got one person to say that they think these last two are anaphase and telophase, which of course they are, and that could probably get you enough to guess that the rest of this is in cell cycle order. But if I scrambled certainly those first four, you would probably be out of luck. I would certainly be out of luck. Computer can do it just fine. Computer can use things like textures that we do not actually have any access to with our crappy human brains, and it can actually make quite nice classification of cells into these cell cycle phases. And when it's making mistakes like S is sometimes confused with G1 or G2, but it's not confusing S with telophase. And this is not a perfectly switch-like process. And so some of this classification error is about that biology is messy, not that our network's actually bad. And so you can do this with deep learning features, you can do this with classical machine learning. And a pretty spinny movie showing these cells all in feature space by cell cycle phase, they cluster, yay. So we can start using that advantage and saying, okay, well, there's things computers know that we just don't know and that make no sense to us. And we can start measuring really weird things about our biological objects, like Zernike polynomials. How much does your object of interest look like one of these weird little circles? And if I tell you that your cell got twice as big, again, you probably have a picture in your brain what that means. If I tell you your Zernike 5-1 polynomial went up by twofold, you probably do not have a picture in your brain. I certainly don't. Maybe your brain works better in Zernike space than mine does. If so, congratulations and please teach me. But if we take these Zernike polynomials and sum them up, there's clearly information here because these are not perfect representations of cell shape, but they're certainly better than we were getting with, something like the diameter and the orientation. We can certainly tell a lot more here. So there's information that's valuable even if it's not human intuitable. Texture metrics are another fantastic one. Again, it's the texture of those bright field cells that the network is really learning and saying these textures are really subtle things that can tell us if a cell is in G1 or S or G2. But our brains simply do not have access to that information. So what is the point of taking measurements that don't make any human sense? Well, that's where we get into, again, this sort of last thing about what are our unknown unknowns in our biology. And so this idea of image-based profiling, I think I have to sell this a lot less than when I started talking about this a few years ago after I joined what was then the Carpenter, now the Carpenter Singh Lab, because people are now used to getting these high-dimensional feature data sets from things like single-cell RNA-seq, and then doing clustering and just sort of seeing what pops out, or spatial transcriptomics, which is also, imaging, but is a different, usually you're doing things like the spatial transcriptomic clustering of your gene counts, which is still not really imaged. But you can take those high-dimensional image features that make no sense to human brains, and you can put them into a clustering algorithm, and you can see if interesting biology falls out. So this is one of my favorite papers from early in the field, and by early in the field, I mean 10 years ago. So here we have a nuclear dye, an actin dye, and a tubulin dye, and we have drugs from known mechanism of action class. We want to say if we take image features of those three dyes, can we take these drugs with known mechanisms of action and put them back together with their friends? You're probably not super thrilled by 67% accuracy until I tell you this is just the nucleus. So we're getting perfect accuracy in protein synthesis, which is not happening in the nucleus. We're getting perfect accuracy on cholesterol lowering, also not happening in the nucleus. If we get 3 channels, 94% accuracy. And again, even the mistakes that this network is making, DNA damage and DNA replication are incredibly interlinked processes. And so to some degree, we may also just be in the regime of we have overclassified things where biology is indeed just kind of messy. And so this has now been put into the clinic. There's now a pharmaceutical company called Recursion-Based Pharmaceuticals that was based basically off of this paper. Anne is on their board. They named a building after her. I'm not on their board and no one has ever named a building after me. But they did this great experiment that I love because I love this picture. Here are cells that have a knockdown of a gene that causes the disease. And they said, can we take our knockdown cells, add drugs, and make them look healthy? And as a microscopist, I can see this phenotype. I can totally see this phenotype. This phenotype is very obvious. And then they asked the computer which drugs make the cells look the most healthy, and they asked the biologists, and then they made drug lists and they gave the drugs to mice. If you are a mouse, you really want the drugs that came out of the computer, because again, our human brains see a phenotype, but we don't see the right one. So the mice do much better on drugs that were picked by the computer. So this led to the idea of, can we just make a really inexpensive, easy to scale imaging assay where we can maximize the amount of data? Basically, how many organelle dyes can we cram onto a standard 4-color microscope, so that we can make these very rich imaging feature spaces where then we can, again, just sort of see if interesting biology falls out. And so if you are a microscopist, you probably recognize some of these dyes. They may be sitting in the back of your freezer right now. We just put out the V3 of this protocol earlier this year. And when I joined the lab was right around the time that the V2 protocol came out, and I was very skeptical that this gives you anything useful at all, that this is really of any use, but I've since drunk the Kool-Aid. If you're not a RAF-RAS pathway biologist, it's okay, neither am I, but you don't really need to be to see that here we have copies of wild type alleles in cell painting feature space now. We've done cell painting of overexpressed genes. We're looking in cell painting feature space and doing some clustering. The replicates cluster, yay. These are all RAF-RAS pathway members, and these are cancer-causing alleles of these genes, and they're in the next cluster over. So in cell painting feature space, we can tell not just that these genes are similar, but that they're not quite the same. And so Mohammed Roban, who now has his own group, spent a lot of time looking at pathway interaction stuff. We found a lot of known stuff, which, you know, when you're validating a technique, yay, but found an anti-correlation between NF-kappa B and Hippo that was not well literature supported. So we asked our wet lab friends, since we are dry labs, to go out and make a reporter assay. And I'm telling you the story, so you can probably guess that, yes, when we have a reporter assay for Hippo and we overexpress NF-kappa B genes, Hippo goes down. So this is novel pathway biology from organelle dyes. There was nothing in this experiment that was set out specifically to detect this interaction, but by anti-correlation in cell painting feature space, we were able to find it. And I just want to sort of emphasize one more time, your brain can't do this. So here are cells that are resistant to bortezomib, proteasome inhibitor, versus sensitive to a proteasome inhibitor. I've looked at these images a lot. I can't tell the difference between the left and the right. I don't think you probably can either if I shuffle them, but the computer actually does a pretty good job of telling the sensitive from the resistant. And we can now start doing this in really fun ways, like combining it with other omics. So we can talk to our friends who take iPSCs from healthy donors and do whole genome sequencing of them. And then we can ask them, hey, can you put some cell painting dyes on those iPSCs? Because it's only about $1 a sample. And then can we do things like look at the feature matrices and see if we can connect genotype to phenotype? And it turns out that when you have 300 patients and 3,000 cell painting measurements and a whole bunch of SNPs, cursive dimensionality hits you real hard. But even after all of that very aggressive correction for multiple testing, we do get a few genes that come out in terms of an individual SNP that connects to an individual morphological measurement. And just to be sure, Matt Tegmeyer, who just left Broad to start his own lab, which I'm heartbroken about, actually CRISPR'd in these particular SNPs and found that they do exactly what you expect in the CRISPR context as well, that these things are not just 3 accidental coincidences, they're actually causative. Now, only one of these 3 is actually a phenotype that a human can even see. The reviewers did not have an easy time with that concept, but here is the one that is human visible, but it's still even only sort of. So this talks about how in the ring directly around the nucleus, how symmetrically distributed are the mitochondria. If you know that's what you should look for, you can actually correctly unscramble these, but if you don't, these are pretty similar looking. Now, why does this gene do this? Absolutely no idea. But now we have a biomarker for this SNP if that's ever interesting. In the last sort of minute or two, I just want to talk about, okay, this is great. So now what can we do with it? So we can take that same thing I talked about with the NF-kappa B and the Hippo, and we can now sort of scale this. We have tens of thousands, and in fact now hundreds of thousands of compounds of cell painting data in the bank, and lots of genes. Can we look for matches and anti-matches? It doesn't work all of the time, but it's data that you essentially get for free and just sort of falls out of screens you've already done. This was a particular microscopy assay called the cell health assay, where you make 2 plates with 2 different sets of dyes, and you say, how unhealthy are the cells based on my drug treatment, CRISPR treatment, whatever you are happening to like to do to some cells. And so we said, well, cell painting's 1 plate. Can we do this in 1 plate instead of 2 plates? And can we then get all the benefits of cell painting for free on top? It's also much less expensive. And, turns out 70% of the time it works 100% of the time. If the feature or you really care about in the cell health assay is down here, you might need some more training as opposed to this very simple regression model. But if you care about one of the things up here, you might as well do cell painting and again, get the extra cell painting benefits for free. And this has now been taken to 11 by Juan Caicedo, who has just now moved to University of Wisconsin, what if you take compound structure information and cell painting information and gene transcription information and you stick it in a blender and you shake. Can you predict imaging, or can you predict imaging and non-imaging assays? Can you predict biochemical assays, bacterial assays, yeast assays? This is, these data are all coming from human cells, but can we predict even in the wrong organism? And again, you can't always get an assay that works at a really high accuracy, but it turns out about 10 to 30% of the time you can. And if you're going to get a result, you're probably going to get it from cell painting. And so if you can do this again and train one of these classifiers off of data that, again, commonly already exists, all of this was done with data that somebody had already captured for a different reason, you can start now to imagine some fun possibilities. So now this is where you're like, okay, well, cool, good for you. You have this giant data bank of cell painting things, like, congratulations, I'm happy for you. What does this mean for me? It turns out that you actually do have access to a whole lot of cell painting images because Aaron Weisbart on my team, Shantanu Singh and Aaron Chu, who just left AWS, unfortunately, got together and helped us put together the cell painting gallery. So this has about 300 terabytes of cell painting images and about 300 terabytes of cell painting measurements. This is all CC0. You can do whatever you want. We would ask that you cite us, please. That would be very nice. It would help convince AWS to keep paying for this. But yeah, you have 600 terabytes of data. Have fun. Among that 600 terabytes is the JUMP cell painting dataset, which we released about a year ago. 117,000 compounds, about 20,000 genetic perturbations. The compounds were sent in this fun, elaborate, didn't cause trouble with customs at all, compound swap scheme, where plates of compounds actually went to physically other locations, so smart people can work on things like batch effect correction. This is all up there, and it took academics and industry. Ann Carpenter is a wizard. She got 10 pharma companies to sign 1 legal agreement to make public data. She is a wizard. We also just put out this preprint after 6 years of working on this project, where we did whole genome pooled CRISPR screens with a version of cell painting that works in pooled CRISPR screening. You have access to all of this data as well. 1 screen in A549 and 2 in HeLa in different media. There's a lot of fun stuff here. We hope you enjoy. So hopefully at this point, you're thinking, wow, this was all really cool. I want to learn how to do this. For the various tools, there's lots of in-app documentation, and we have websites that, again, have lots and lots of documentation where you can learn lots of things. If you want to do this image-based profiling, we have a whole handbook for this. And we put out a current protocols paper earlier this year about how you actually interpret these clusters once you actually get them out. What's actually that last step of turning the clustering into biology? And no matter what you're doing, bioimagingguide.org is something that we did alongside Bioimaging North America's Image Informatics and Training and Education working groups this year. This is the guide to what you don't know about quantitative microscopy. So whether you are a data scientist and you've never touched a microscope and you want to learn a little bit about what don't you know at the beginning of the pipeline, or whether you are a microscopist who's never touched a computer and you want to know the other half, we have a nice Jupyter notebook or Jupyter book workflow where you can do things like open and collapse different software tool information, and we can put notes in the margins, and you can learn about things like what's going wrong. And it's not a place that's going to teach you everything, but it's going to teach you what are the concepts you should know and point you at resources of where to go learn them. We're now translating this into 8 other languages. Again, I mentioned this checklist paper that Quarep Limi put together in terms of if you're publishing image data or image analysis data, please actually look and see how to actually do that in a way that's going to be helpful to the community. Also, we have a fun little Jupyter book there. I'm still trying to get these checkboxes to work. They're being very annoying, but they will be checkable eventually. And forum.image.sc is where about 70 different open source image analysis tools all hang out and have one common help forum. So you can say, here's my image, here's my question, someone, dear God, help me. And you have experts from 70 tools who will tell you which is the right one and how to use it. And it's an incredibly friendly place where just people love being dorks and talking about, oh yes, let me write you a huge script to help you analyze your question because I just love helping people. It's a really actually beautiful space. That is funded by the Center for Open Bioimage Analysis, which we are part of. We make software and do projects in concert with biologists and do things like then go tell people about. Our team has office hours if you want to talk about this stuff, but you guys have the Image Analysis Collaboratory in the building, so you may not need us. But if you want to talk about lots of image analysis with lots of people who really like talking about image analysis, with Simon and I, we've recently started Boston Bioimage Analysts. You can join. We have a Google group. Website is in progress. And so with that, I talked about a ton of work from a lot of really smart people. Most of them were not me. Most of them were these people and alums of the 2 different labs. These are our tools. These are our resources. And thanks. Have time for a few questions.
Thank you, Beth. I'm going to start by reading one here, saying great talk and so on. My question is specific. How can this be applied to complex cells like neurons, where we want to monitor both the somatodendritic and axon that are so different in area and volume? How can you do segmentation in these cells in its entirety?
Yeah, great questions. The questions around segmentation of neurons. Almost nothing does that well automatically yet. There are a lot of things working on it. You know, the Genelia Flybrain people, we're working on some tools that we hope are going to be user friendly. Nothing is really great at it yet, but I'm hopeful in a year or 2 I can give you a better answer. In the short run, you can come talk to us and we'll help if we can.
Thank you for the talk. It was fantastic. I had a question about how we go from, I guess, identifying what features are relevant, because some of the features are unknown unknowns. And if you're interested in applying the morphological profiling to your cells and data set of interest without using a cell painting assay, how do you identify what features to even include in the first place?
Yeah, that's a great question. So how do you actually sort of figure out features? How do you then connect them to biology? Is the short version is, how do you identify which features to do? Our lab motto is measure everything and ask questions later. So if you're using something like Cell Profiler or Skimmage or whatever you're using, just stick on as many measurements as you can possibly manage to fit inside your data frame without it exploding. Disk space is cheaper than repeating the experiment, typically. And in terms of then, how do you actually figure out what is the biology there? This is a whole paper we have on that. It's hard, and sometimes you never actually figure out what actually that feature means or how it connects or what's the next experiment to do based on the features you found. Usually you get luckier than that, but occasionally sometimes you don't, which is always a sad day. But we have some techniques on how you can get started and how you can try.
Sort of latch on to that question. So measure everything, and indeed, Cell Profiler has very many features you can extract. Obviously, you don't want to measure all Harlech features, for example, because that's computationally very, very expensive. But is there any work on developing new features, maybe inspired by whatever a neural network is looking at when it's classifying, and sort of putting in things that are not based on classic image analysis, but just pure heuristics?
Yeah, that's a great question. So new kinds of features, not just things like Harlech features, which are, if you've never heard of a Harlech feature, don't worry, high-level shape descriptor or texture descriptor. So there is such a thing as Deep Profiler. It's basically based off of ImageNet and the idea of basically sort of doing classification and then getting, using a feature layer of your neural network to then make deep learning based features. There's a lot of work in the space. A lot of stuff says that the classical features are better. A lot of it says the deep learning features are better. We still don't really know right now as much as we can. We're taking both and just sort of trying to see what for a particular problem works better. I imagine with time, the deep learning features for pure classification will probably get better than the conventional ones, but because they're not really interpretable, there's folks like Asif Zaritsky, of course, doing clever things around deep learning, interpretable neural networks for biology, and lots of people doing it outside of biology. But because you want to know what's that next experiment to do and what is the feature that's actually driving the difference in your experiment, I don't think conventional features are probably going to go away anytime soon.
One last
online question. How do the tools do with segmenting different kinds of objects when there's a mixture of objects and then analyzing features of the specific type of object of interest?
Yeah, that's a great question. So different kinds of objects. Different tools are going to do really variably at sort of different kinds of objects. So I showed here Coco, which is not truly an instant segmenter, but is an object detector. Coco has a few hundred classes built in. And so there are certainly networks and tools out there where you can very easily get lots of classes of object right away. In a cell profiler pipeline, you can keep adding object segmenters and get lots and lots and lots of kinds of objects. And then it's very easy once you have objects to just sort of plug measurements onto them. How easy that is in your favorite tool is going to depend a lot on your favorite tool. And it might be easier to actually just identify all objects as cells and then classify them into subsets than actually trying to find your different subsets with segmentation right up front.
Beth, thank you so much. We're going to stop here. People that want to ask more questions can hang around, of course, but otherwise we thank you.